morning students, welcome to B.Sc-II Zoology class. So now I am going to discuss the second phase in gastrulation of chick embryo. Gastrulation in chick embryo. Okay. So there are three phases that occur during the gastrulation in chick embryo. So first phase we already discussed, formation of endoderm. Formation of endoderm is completed. How the endoderm is formed? So endoderm is the first germinal layer that is going to be developed during the gastrulation. Endoderm becomes the wall of the elementary canal of the embryo. So the endodermal cells come from blastoderm. They come from blastoderm. Blastoderm is nothing but epiblast. From the epiblast, cells are moving downward to form hypoblast. So hypoblast cells are nothing but endodermal cells. It is going to be developed into the elementary canal. Okay. The cavity present between hypoblast and yolk is known as the orchentron. The orchentron, roof, lateral walls and the floor of the gastrocele, the orchentron is lined by endoderm. So endoderm is developed from hypoblast. Hypoblast is nothing but cellular layer present below the epiblast during the gastrulation. During gastrulation, three processes occur. The first process is formation of endoderm. The second process is formation of the primitive streak and mesoderm. And the third one is formation of embryonic axial and paraxial structures. Okay. So the first phase is completed. Now let us talk about formation of primitive streak and mesoderm. Formation of primitive streak and mesoderm. So what is primitive streak? Look here. So this is what the blastodisc where embryo development occurs. So this is blastocell. I told you when the egg is laid by the hen, the embryo would be in blastula stage. Blastula. That blastula, when you observe from the upper surface, it would be looking like disc shaped. Therefore, that blastula is called as discoblastula. Disc-like blastula is there, and this blastula area embryo has two areas. This disc shaped structure has two areas. Central part is transparent, almost translucent. This is known as area pellucida, and the peripheral, somewhat opaque region is known as area opaca. Area pellucida, and this is area opaca. Because the epiblast at this region is connected to underlying yolk. Whereas here, this epiblast is not connected to underlying yolk. In between yolk and epiblast, there is a cavity, and that cavity is filled with liquid. Therefore, this part is looking like transparent. Therefore, this transparent region of the blastodisc is referred as area pellucida. In the middle region of the area pellucida, narrow strip is appearing, and this strip of thickened epiblast due to the inward movement of epiblast cells at this region streak is formed. The thick line is developed along the midline of the area pellucida, and this is known as primitive streak. What is this? Primitive streak. So primitive streak is nothing but thickened strip. This straight strip of thickened cells coming from the epiblast cells, which is moving from posterior end towards anterior end, and that midline, thickened midline of the area pellucida is called primitive streak. This primitive streak is formed due to the inward movement of epiblast cells, and at the center these cells are aggregated to form thick strip-like, band-like, cord-like structure. That cord-like strip, thickened strip, is known as primitive streak. This begins to start from the posterior edge of the area pellucida, and extends up to 3/5 part of the area pellucida. It is not reaching the anteriormost edge of the area pellucida. Now it's not meeting that one. It moves up to 3/5 part of the area pellucida. Okay, so this is what primitive streak. So primitive streak formation is very important for the formation of what? Formation of notochord, nerve cord, the mesoderm. All regions start to appear from the primitive streak after the formation of primitive streak. So this is primitive streak. How the primitive streak is developing? At the beginning, this is what area pellucida. It is disc-like, whereas here it is oblong, bit elongated because of the development of primitive streak. Before the formation of primitive streak, the blastodisc would be like this. The blastodisc can be differentiated into two regions: area pellucida and area opaca. Okay, here at the posterior edge of the area pellucida, you can see the accumulation of cells. These cells are coming from the epiblast or blastoderm, upper layer of the cells. They are coming towards the posterior mid region of the area pellucida here. As a result, thickened and wider part of the blastoderm is seen at the posterior edge of the area pellucida. So this is the beginning of primitive streak with a thickened blastoderm. Here the blastoderm is thick. It is thick. Okay. Gradually it extends forward. Can you see this accumulation of cells? Look here. The cells are moving this side. Can you see? They are moving inside, going inside the mid region. So, as a result, this line is thickened due to the accumulation of cells that have come from blastoderm. Except this region, here and here, the layer is very thin. It is not thick. Because it is not multilayered. Whereas here it is multilayered. Several cells are accumulated here to form cord-like structure just below the epiblast, and that is called primitive streak. And this primitive streak extends forward up to 3/5 part of the area pellucida. Okay. So next, what happens now? Look here. So here at this region, cells are separating. So first, the primitive streak would be like It's a straight line, thickened part, like this. So this is what primitive state. So this is what epiblast at the midline of the area pellucida. This blastoderm is thickened due to accumulation of blastodermal cells. Now what happens? Gradually this side cells they separate and move downward towards hypoblast. Towards what? Hypoblast. What is this? This is what? Hypoblast. So hypoblast is nothing but formation of hypoblast we already discussed. For the formation of hypoblast, several theories are put forth by different people. We already discussed. So this is what? Hypoblast. But hypoblast is going to be developed into endoderm. So these cells at this region, the cells are moving separately, singly. Look here. During gastrulation in the birds, the cells move downwards singly. They move singly. As a result, what happens here? The cells number is less here. It is making groove-like structure along the primitive state, midline of the area pellucida. So earlier it would be like this. As the cells are moving, separating from here, separating from the epiblast, it curves like this. It curves like this. Due to the involution of cells, these cells are separated. Not all cells are, all cells are not following the same cell. Okay? As a layer is not moving. Cells are separated. These cells move downward towards the hypoblast. As a result, groove is formed. Groove is formed along the primitive state. That groove is called as primitive groove. What is this? Primitive groove. Okay? Primitive state formation is completed. Okay? Now what happens now? The cells of this region are moving downward. They are moving downward singly. As a result, groove is formed along the midline of the primitive state. Okay? Groove is formed. This is elevated. Groove is formed along the entire length of primitive state. At the anterior end of the primitive state, the anterior end of the primitive state is thickened. So this is called Hansen's node. Hansen's node. Due to the immigration of the cells, the single movement of cells singly towards the hypoblast and laterally to form the mesoderm is referred as immigration. During gastrulation in the birds, the cells move downwards singly. This type of gastrulation movement is called immigration. Due to immigration, what happens? Earlier, when primitive streak is formed, the surface of the epiblast is even. It is smooth. When immigration starts, along the primitive streak, primitive groove is formed. Primitive groove is formed due to the development of primitive folds, two folds, this side and this side. Folds are formed. Fold formation continues till the Hansen's node. Okay, so this is primitive ridge, primitive groove, Hansen's node. Hansen's node is anterior tip of the primitive state, head part of the primitive state. That head is thickened. That head part of the primitive state is called Hansen's node. Now, due to immigration of cells, there funnel-shaped pit is formed. This is called primitive pit. Funnel-shaped pit is formed. So these cells, okay, the cells that are present in the primitive pit or Henson's node region, they are going to be developed into notochord and nerve cord. Notochord, nerve cord, mesoderm. Okay, so the primitive groove or the funnel-shaped pit in the primitive groove is developed due to immigration of cells. Immigration of cells. Thus, the primitive state is completed. Primitive state formation is completed. After the formation of primitive state, primitive groove is formed. At the anterior end of the primitive groove, primitive pit is formed. Primitive pit is formed. Okay. So later on, after the formation of primitive state, what is developed? Next process, the formation of embryonic axial and pyramidal structures. Axial and pyramidal structures. So immigrated cells, they move towards the hypoblast and laterally they spread to form mesoderm. Okay, during the formation of primitive states. Next, during the formation of embryonic axial and pyramidal structures, the primitive states, length is becoming shorter and shorter due to the development of notochord anterior to Henson's node. The caudal cells, notochordal cells, they come from Henson's node. Primitive pit. As the notochord development begins, size of the primitive state regressing, shortening. So that how it happens, we'll see in the next video. Thank you.